A-Level Biology Edexcel Enzymes active sites, factors affecting activity and denaturation. Concept first, then real exam questions. Let's begin with enzyme action and active sites. Enzymes are biological catalysts. They speed up reactions, but they are not used up. First, the substrate approaches an active site with a complementary shape. This shape gives the enzyme its specificity. Next, the substrate binds to the active site, forming an enzyme-substrate complex. The reaction then produces products. These products leave the active site, and the enzyme remains unchanged. It can be used again. The key memory point is simple. The active site controls specificity, but the enzyme is not used up. Now let's look at factors affecting enzyme activity. As temperature rises, particles gain kinetic energy. Successful collisions become more frequent, so the rate increases towards an optimum. Above the optimum, bonds in the enzyme can be disrupted. The active site changes shape, and the rate falls. Enzymes also have an optimum pH. Extreme pH can change the bonds holding the active site in shape. Increasing substrate concentration raises the rate while active sites are available. When every active site is occupied, the rate levels off. Always connect the graph to successful enzyme-substrate complex formation. Finally, let's explain denaturation. High temperature or extreme pH can disrupt the bonds that hold an enzyme in its three-dimensional shape. In the normal enzyme, the substrate fits the active site, so binding can occur. After denaturation, the active site has changed shape. The substrate no longer fits. Fewer enzyme-substrate complexes form, and the reaction rate falls. Denaturation is usually permanent because the enzyme's shape has changed. In an exam, link the changed active site directly to the loss of successful binding. Question 3AI. Lipoprotein lipase is a biological catalyst. Which statement correctly describes its effect on the rate of reaction? Option A, it decreases the rate by increasing activation energy. Option B, it decreases the rate by reducing activation energy. Option C, it increases the rate by increasing activation energy. Option D, it increases the rate by reducing activation energy. Take 1 or 2 seconds to choose the statement that matches the definition of a catalyst. The answer is D. A catalyst increases the rate of reaction by reducing the activation energy. It does not provide extra energy. It makes an alternative pathway easier. Question 3BI. Some people have a mutation in the gene coding for lipoprotein lipase. Give 2 reasons why the information in the table does not support the suggestion that the mutation increases cardiovascular disease risk. Read the table by comparing people without the mutation with people who have it. Total cholesterol is lower with the mutation. LDL cholesterol is also lower, while HDL cholesterol is slightly higher. Now pause and identify 2 comparisons that do not support an increased cardiovascular disease risk. The 2 answer points are: total cholesterol is not higher in people with the mutation, and LDL cholesterol is not higher, while HDL cholesterol is not lower. Question 4BIY. Flatworms are often found in water at about 15 degrees Celsius. Why might this temperature be favorable? At this temperature, there is still plenty of dissolved oxygen in the water, creating a concentration gradient for oxygen to diffuse into the flatworm through its body surface. Enzyme activity also depends on temperature. Below 15 degrees, enzymes and metabolism work less effectively, so the animal would have less energy for life processes. Putting those ideas together, 15 degrees is a balance between oxygen availability and enzyme activity. Here are 4 scoring tips for enzymes. First, name the active site precisely. Link rate to enzyme-substrate complexes. Explain optimum temperature and pH. State how denaturation changes shape. Link shape, conditions, and rate to secure marks. Keep practicing real exam questions on EduNinja. Use topic practice. Follow step-by-step solutions. Build smart paper practice. Use revision notes and more.